All right, hello class. We're going to go over this worksheet that we did the other day in class. So it's just a basic practice worksheet for the monohybrid cross as well as some good vocabulary practice. So let's start with our vocabulary. So as we run through, I'm just going to show you everything. This is a genotype made of the same alleles, okay? So that means we have two copies that are exactly the same. And we talked about this in two ways. So this could be, okay, for example, if we had a trait that we were following with the letter A, it could be the same that they have two copies of the dominant trait, or the same that they have two copies of the recessive trait. And remember, just the prefix um, that means the same is homo. So this goes along with D, homozygous. Then two different forms of genes for a single trait. So that is what goes along with the alleles. So we could have for example, a gene that says we're going to have the hitchhiker's thumb, we're not going to have the hitchhiker's thumb. So those are two copies of the same type of gene. So both telling us about how the joint in our thumb is going to work. 3. Gene that is always expressed. Okay, this is the this is the one that I feel like the wording is a little bit unusual, but that's okay. B, so dominant. A dominant trait will always be expressed if present. So I'm going to add a qualifier. So if present, it'll always be expressed. So it doesn't matter if you're, for example, AA, so homozygous dominant, or heterozygous. So if we're looking at that dominant recessive relationship, then if you have one copy of the dominant allele, remember going along with the capital letter, then that's what will be expressed. Next one, the gene that is expressed only in a homozygous state. So this means, okay, it's not going to be expressed all the time, but only homozygous, and we can assume that one would be recessive. So that's the only thing we're going to see if we're homozygous recessive. Okay, so a person who is heterozygous will not show this recessive trait. It'll be hidden by the dominant trait. 5, genotypes made of two different alleles. So two different, okay? So now we have the capital and the lowercase. And then the prefix hetero, we said also means different. So that's going to go with C. All right, so now we did this practice. And some people struggled with the practice as well, so it's good to take a look at them and just review. So looking at the dominant alleles, this equals, okay, so big or capital letters. Okay, recessive alleles was a small or lowercase letter. Okay, so we should circle all of the options available. Homozygous dominant, so homo meaning the same, okay, dominant meaning capital letters. So we're circling all of our double capital letters. Homozygous recessive, so homo meaning the same, okay, recessive referring to the lowercase letters. So now, well, it looks like I'm a little bit crooked up there. Let's see. All right, there we go. So now we're circling all of our double lowercase letters, so hopefully I got all of them on there. The genotypes in which the dominant gene must show. So if we're just looking at the dominant recessive relationship, we're looking at all of the possibilities where they have at least one copy of the capital letter. So we have all of our homozygous dominants as well as our heterozygous, because the heterozygous will show the dominant characteristic that goes along with the capital letter. Then genotypes in which the recessive gene must show. So remember, recessive now, so we're looking for people who only have that option. Okay, so they only have the lowercase letters. So we find the ones that it applies to. So I just found these two, so lowercase a, and lowercase r, and I circled them. Sorry, I'm drinking my coffee. Then Punnett squares. Okay, let me see. I'll move this, shift so I have a little bit of space. So first one, we're looking at the ones that are correct, all right? So I found the first and the last one looked to be correct to me. The other two have errors, so it just looks like as they were bringing the letters or the two alleles across, they made some mistakes. So this one, we should have brought the big D down and the lowercase d across to have a heterozygous genotype. And the same here, they should have brought the lowercase letter down and then the capital letter across, so you get a heterozygous genotype, and they didn't get it. So that was the first mistake I caught, so I just made a note and I circled the others. Then looking at what the Punnett square sets up again. So what are the letters outside of the square? So that should be what's on the side and the top. Well, those represents the genes of the parents, so the genes that could exist within their gametes. Okay, so the options that the parents could give for genes for their children. Okay, and I made mine. Oops, oh, mine is two-sided. I wasn't sure. Okay, so then I'm going to zoom to look at the top. Okay, next one is what is inside the Punnett square. So it's the possible gene combinations of the offspring. Remember, it's just probability, so it doesn't give us any guarantee of what the offspring will be like. Okay, it's just a probability thing. So next, we're trying to just bring the letters across for the next one, so we're just practicing. And then it asks us to shade them in, but I do not have a bunch of different colors on me, so I didn't do that. But I'm going to hopefully walk through at least a couple with you. So if we look at the homozygous dominant, the heterozygous, and the homozygous recessive. So let's see, it said to mark them red. So homozygous dominant should be red. So there's none here. Okay, this one should be red. I'm just going to mark with a lowercase r. That one should be red. This one should be red. This one should be red. Okay, and that's all I see at the moment. Okay, so heterozygous should be green. So this one green, green, green. So this whole thing should be green. This should be green. This should be green. Okay, this should be green. This should be green. Okay, and then again green, green. And then all of the homozygous recessive ones, it says leave unshaded. So then we're looking at this one, and this one. Okay, and you'll actually, if you do enough, you'll see a pattern that starts to form. So the ratios that come up as the possible outcomes for the children will be very similar once you practice them a lot. Okay, then we're looking at guinea pigs. And we're talking about guinea pigs that could have short hair or long hair, and short hair is dominant over long hair. So setting it up, it's telling me a little bit about my guinea pig parents. So it says we start with a guinea pig that is heterozygous and one that is homozygous recessive. I have no idea which one's mom and which one's dad, so it totally doesn't matter. So I just decided that I was going to put the heterozygous one on the top and the homozygous one on the side. I did a little slash because it's hard to tell a capital and lowercase s apart. So I just did uppercase, lowercase. Okay, lowercase, lowercase. Put them together. Okay, then I'm counting them up. So it says expected number of offspring. So we have the short hair, we have 1, 2, and long hair, 1, 2. Okay, so a ratio is 1 to 1 if we reduce it. So this is like a 50-50 chance, okay? So there's a 50-50 chance that they'll have long hair, half and half, whatever you want to say, that they'll have long hair to short hair. Then now both of the guinea pigs are heterozygous for short hair. So now we set up a little bit differently. So we have two heterozygous, one, okay, 1, 2, so different alleles, and then we bring them together. So now our genotypes are different, right? So we have, because we're looking at our phenotypes, the characteristics that they actually show, but we have a homozygous dominant, we have two heterozygous, and we have one homozygous recessive. But the outcome is going to be that we have 1, 2, 3 with short hair, and 1 with long hair, okay? So this time, the probability of them, the ratio would be 3 to 1, that they're short hair to long hair, or if we're just looking at it, because there's 1, 2, 3, 4 possibilities, there's a 75% chance that they will have short hair. Okay, and this is short to long. All right, perfect. So then we set up another one at the bottom. So now we're looking at hornless and horned cattle. Okay, so it says our bull, which is our male, mated with a homozygous horned cow, which is our female. Again, the position of the male and female on the end will not impact your results at all, so if you switch them, it doesn't matter. But our homozygous dominant, our homozygous horned bull, yeah. should be, or hornless bull, sorry, I'm getting confused. So should be capital H, and then it's crossing with a lowercase h, lowercase h. So we have homozygous dominant and homozygous recessive. So then we set up our Punnett square to look at our outcomes. Okay, so we're going to have dad at the top, mom on the side, and we have 100% the same. So if this is our mating cross, we're always going to get heterozygous children. So when we look at the bottom, if we set up a ratio of what we're looking for, if we say we're looking for homozygous dominant to heterozygous to homozygous recessive, we'll get, and I made a little scribble because I was moving too fast. I went 0 to 4 to 1, so that means 4 are going to be, H, capital H, lowercase h, or heterozygous, which is 100% hornless. So we're just analyzing it super quickly. Then we're looking at tomatoes and whether or not we have red or yellow tomatoes. So let's take a look at that. So this is the same situation where we have, I believe, a homozygous red tomato and a homozygous yellow tomato. So we're going to see the same types of outcomes. Okay. So we set it up. Again, homozygous dominant, homozygous recessive. Every time we cross, we're going to get 100% heterozygous. So we write out our information. We're going to write out our ratios. So we have 4 that are red, that are heterozygous, and that's 100% of our tomato plants. Now this one gets a little bit interesting because we started labeling who our parents are and who F1 were. So this is our first filial generation, or these are like our parent tomato plants. These is the first generation of offspring of the tomato plants, and F stands for filial, so that means offspring or kids, right? And then we're taking the kids and we're saying that the offspring or the kids are reproducing together to form a new generation of tomato plants, which is not as creepy when we're talking about tomatoes as it is when we're talking about people. So let's say we take our first generation results. Now we're crossing them and we're going to make different tomatoes. So we're taking a heterozygous individual with another heterozygous individual. We're setting up our Punnett square here, and now we're seeing a different ratio of outcomes. So we're looking for our genotypes and our phenotypes. So we have 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. On the side, if that's what I'm writing out that I'm looking for, okay, these are my genotypes. So I have my 3 genotypes. We usually go from the homozygous dominant to heterozygous, homozygous recessive. I write out 1 to 2 to 1, and then I'm looking at my phenotypes. So what are the colors of the tomatoes? Like, what do they actually look like? So I have 3 red, 1 yellow. Okay, perfect. Then we're looking at this. Humans and the tongue rolling ability. So we said, oh no, my camera. Okay, give me just a second. Let me pause it. Oh, the camera fell off here. All right, sorry, I was trying to adjust my camera and it fell off the stand. So next we have a non-roller. So he's homozygous recessive, and a roller, but she's heterozygous. So we put the information there. And we're just trying to set up the cross. So that's all we're doing. So they're giving us the genotypes and the phenotypes just by reading the information. I set up my cross, and it's asking what is the probability of the couple having a child who is a tongue roller. So looking at our setup, we have a homozygous recessive and a heterozygous. So these 2 are the rollers, and these 2 are the non-rollers, if we want to put it that way, non-rollers. So it's 1 to 1 or half or 50%. Okay, either any way you write it, that's what we're looking for. Then the last one is kind of analyzing the family to figure out the genotypes of the parents. So we know we have brown eyes dominant over blue eyes, so I made a little note on the side that brown would go with capital B and lowercase b would go with blue eyes. So we know that our man has brown eyes, I believe, and our woman has brown eyes, but both of them had a blue-eyed parent. So I actually set up a little pedigree. So we have our male and our female. They're going to have a kid, and we want to know, like, what's the chance that their child will have blue eyes. So knowing that, okay, each of them have a parent with blue eyes, they have to be homozygous recessive. Okay, the other parent, we're not totally sure. They could be heterozygous or a homozygous dominant. but the genotypes of the two parents, okay, so of the people we're concerned about, they have to be heterozygous because they have a homozygous recessive parent. So this person could only contribute a small b, okay, and they must have gotten the other b for the brown eyes from the opposite parent. Okay, so capital B with brown eyes from the opposite parent. So then we're wondering what's the outcome of their children going to be. So then we're going to set up our Punnett square again. So we have our our heterozygous and another heterozygous. We get one homozygous dominant individual, two heterozygous individuals, and one homozygous recessive individual. And I set up my ratios here, so just looking at genotype. So remember, this is genotype. And this is our pheno. I don't have room, so I'm just going to write pheno. So phenotype. But really only one of the four could potentially be blue eyes because we only have one homozygous recessive option. So when I write that out, I'm just going to write, okay, a quarter of them could be blue-eyed, or 25%. Done. All right, so hopefully that helps, and I will see you later.